So, welcome back. Today we are on the side of the room rather than the front of the room. We're going to see if that helps improve the acoustics at all. We do some of that echo that we have going on in this space. It's large, it's empty, it's sad and lonely, and that makes it very echoey. But today I will be discussing cells. So obviously you guys are familiar with cells, right? You learned about cells often in your lives. I'm sure you learned about them in middle school and you learned about them in Bio 1. We're going to delve a little more deeply into the functions of the individual organelles in this class. We won't spend too much time learning about too much of the nitty-gritty, but we need to get a general understanding and a general conception of how everything in the cell works together to make the cells individually unique, and how, for instance, a cardiac cell is different than a muscle cell. Again, I always use that example. That's so dumb of me. A cardiac cell is a type of muscle cell. So let's take it back. Right, so we're going to see how things are different, like a neuron is different than a cardiac cell. Okay. So, starting with, of course, the tenets of cell theory, because we can't talk about cells without cell theory. So cells are the smallest unit of life, meaning that anything smaller than a cell is not in and of itself alive. So like the ribosomes aren't alive, and the mitochondria aren't alive, and the nucleus isn't alive. But that cell is in and of itself alive. So my eyeball cell is alive. All living things are composed of cells. And all cells are derived from other cells, meaning that there's no spontaneous generation of life. So I can't just have a cell poof up out of nowhere. I need a cell to make other cells. And so that's how we've come down the course of evolution, from that original cell to all the cells that we have now. They've all come through other cells, with the exception of the very, very first cell, which was like a magical, crazy, improbable event, but it happened. And once it poofed up into the world and it started replicating itself, right, we've had a never-ending line, a chain of cells ever from that individual, unique, first, original cell. So we're going to talk about some cellular stuff now. So, first topic that we're going to look at within the cell unit is prokaryotic versus eukaryotic cells. So, prokaryotic cells are the more simple version of cells. So the original cells that showed up in existence were forms of prokaryotes. Throughout the course of evolution, even these simple prokaryotic organisms have become much more complex than their original forms. So the bacteria that exist today are prokaryotic. They are really simple compared to us eukaryotic organisms, but they have a lot of complex qualities. They have a very strong ability to adapt and evolve. Over the course of the eons that they have existed, they have developed mechanisms that allow them to do very complex things with their simple little tiny existence. All prokaryotic organisms are single-celled. The nature of their existence does not allow them to become multicellular. The defining feature of a prokaryotic organism is that it lacks membrane-bound organelles. And that characteristic is extremely limiting to the amount of complexity that it can occur within the prokaryotic cell. So the only membrane they have is their plasma membrane, the one that surrounds the entirety of the cell, the cellular membrane, cytoplasma membrane. That's the only membrane within the prokaryotic organism. Whereas the eukaryotic organisms have membrane-bound organelles. So they have these little pockets that are surrounded by little membranes that have unique functions. So these are the organelles that we usually think of. Things like the mitochondria, the nucleus, the Golgi apparatus. All of those fancy little organelles have their own membrane. They're these little tiny compartments of specialized function. And eukaryotes have those. Prokaryotic organisms do not have any of those organelles. And without those organelles, they lack the ability to become specialized cells, and they lack the ability to become multicellular organisms. So prokaryotic organisms are much smaller than eukaryotic cells. So an individual bacteria, its entire existence is smaller than a single one of your cheek cells or eyeball cells or liver cells. So these eukaryotic cells are much larger, even though they in themselves are not organisms, because the eukaryotic cells can compartmentalize their functions through these membrane-bound organelles. That doesn't mean that the prokaryotic organisms lack the ability to do specialized things within themselves, right? So some bacteria have specialized regions, and they wrap those regions in protein. So they don't have a membrane, but that region is kind of bound down here. And so maybe this is like the waste digesting region. And so the enzymes that digest the waste are going to be more concentrated in that area than there'll be other places in the cytoplasm of the prokaryotic organism. But the enzymes are still going to flow around into that cytoplasm because it's not wrapped up entirely. Another way that prokaryotic organisms compensate for the lack of organelles, right? So they need some membrane space to do some work. A lot of prokaryotic organisms, is it a lot? Is it some? A fair amount of prokaryotic species have folded plasma membranes, and that allows them to emulate some of the functions that we see in the eukaryotic organisms. Emulate's not really the right term because they're really the origin of some of those functions, right? And so it's a primitive version of what eukaryotic cells do. And photosynthesis is a prime example of this. So we have photosynthetic bacteria that are able to process sunlight and create sugar. Same way that plants do. Plants use their chloroplast to achieve that, which is a membrane-bound organelle. Prokaryotic organisms don't have that chloroplast, but what they have are these highly folded regions of their plasma membrane where they do those same chemical reactions. So the prokaryotic organisms can achieve some of those same complex functions that eukaryotic organisms achieve through folded membranes or protein-boundish regions. Okay. So main breakdown, prokaryotic organisms are simple. The only existing prokaryotic organisms left are bacteria, archaeobacteria, bacteria. The eukaryotic organisms are much more complex. They're multicellular, most part, right? There are unicellular eukaryotic organisms, but for the most part, we're multicellular. And so these are all the other forms of life on the planet: animals, plants, fungi, protozoa. And the major difference that allows for that is that eukaryotic organisms have membrane-bound organelles, which allow for a much higher level of specialization through compartmentalization. And so the eukaryotic cells can work together to do different things and make this big, beautiful organism. Okay? So, a closer look at our prokaryotic cousins, the tiny little cute little bacteria guys. And we're going to look at what they have. So, again, they don't have any membrane-bound organelles. So none of these things are wrapped in membranes. What they do have is a plasma membrane, which is the cellular membrane, the outside membrane, and it is a phospholipid bilayer, just like ours. But that's the only membrane they have going for them. Within the cell, there's a region called the nucleoid, and that's where the DNA of the bacteria, of the prokaryote, hangs out. And it is defined by a protein fence, effectively. And so it is like regulated about where it hangs out, but it's not isolated from the environment of the cell. So anything inside of the cytosol that's floating around can get to the DNA of the prokaryotic organism. Whereas our DNA is protected by a nucleus, so the environment of our cell doesn't impact directly the contents of our DNA. So the nucleoid is a region where the DNA hangs out, but it doesn't really protect the DNA. Prokaryotic organisms do have ribosomes. Ribosomes are absolutely essential for the production of life. Right? That's where the proteins are being made. And proteins are the way that DNA is expressed. And without proteins, we don't have any regulation mechanisms. We don't have any of the systems or mechanics or structures that we utilize to stay living things, right? So you need ribosomes to make proteins. The things about ribosomes is that they're not big organelles. They're not fancy organelles. They're very simple protein and RNA structures. And they're tiny. So all living things have ribosomes, including prokaryotes. Most prokaryotic organisms have a cell wall that is made out of carbohydrates. So it's a rigid, stiff carbohydrate structure that wraps the prokaryotic organism, that keeps them safe. Because they're just this tiny little cell. They don't want to get squished by the world. So they have this nice cell wall to keep them protected, keeps their shape the way it's supposed to be, for them to function the way that they need to function. That cell wall is very often covered in another carbohydrate complex that's sticky. And so a lot of these bacteria have sticky outer layers on them. And they use that to adhere to different things. Right? So bacteria like to adhere to surfaces often. And they use their sticky layer to do that. Also provides them a little bit of protection from the environment. So if like there's a bad thing that comes in, it can stick to their outer layer rather than getting into them and hurting them. There's the cytoskeleton, composed of microtubules, protein structures, and that includes flagella, the big long taily things that they have off their backside, helps them move around. Right, so not all bacteria have flagella, but the ones that do can move through liquidy things. They can swim pretty much. Or they have cilia, which are the tiny little structures, like hair-like projections off of the cells, and they use those to kind of like crawl around things. And obviously, it's not as great as limb crawling, but like they can move. Again, the cytosol is the jelly that fills the cell. So you get this jelly substance. It has proteins in it. It has enzymes in it. Enzymes are a type of protein. It has sugars in it. It has nutrients in it. All that stuff. The jelly of the cell is the cytosol. Right? The space between basically everything combined inside of the cell is the cytoplasm. Right? And so from wall to wall, inside the cell is the cytoplasm, including the organelles and all the good stuff in there. The cytosol is the actual jelly. And the prokaryotic DNA is a single circular chromosome. And so we have linear chromosomes, we have a bunch of them. The DNA has a big circular chromosome with all of its genes in it. So the full genome is this circular thing. They very often have these little pieces of DNA floating around that they can use for different things, but their primary chromosome is this one big circle. Okay? prokaryotes. So, on the board behind me, I have a list of eukaryotic structures. Now, all of these words should be familiar to you from Biology 1. These are all basically the organelles within the different eukaryotic organisms, and now some of them, such as chloroplast, are specialized organelles that are only found in some types of eukaryotic organisms, plants in this instance. But overall, these are the organelles that are found within the eukaryotic living things. So we have the nucleus, where the DNA lives. We have the mitochondria, where ATP is produced. We have the endoplasmic reticulum, which is a big foldy membrane structure close to the nucleus, right? And so there are two types. There's the rough ER and the smooth ER. The rough ER possesses ribosomes, and so that's where proteins are primarily synthesized. The smooth ER does not have ribosomes. It is responsible for the processing of those proteins as well as lipid production. We have the Golgi apparatus, which is another membrane structure. This one is independent. It's over down here. And it further processes proteins, gets them ready for the world. We have the ribosomes, which make proteins. We have chloroplast, which do photosynthesis. We have vesicles, and there's a bunch of different types of vesicles that have specific functions, but they're basically just little bubbles of membrane floating around inside the cell, and inside the bubble there's different things. And depending on what you find inside the bubble, the vesicle can do different functions. We have vacuoles, which are a different structure of bubbles. They hold liquids. In plants, there's a thing called a central vacuole, which is very important to the structure of the plant. There's the cytoskeleton, which helps maintain the shape of the cell. It helps with motility of substances within the cell. And parts of the cytoskeleton play a huge role in the mechanics of the cell. So there's a thing called a centrosome, and it regulates the movement of stuff during mitosis and meiosis. That's part of the cytoskeleton. We're going to have a little mini lecture specifically devoted to just the cytoskeleton because it is a complex thing, and it's definitely an AP Bio thing. Right? It's not something you would have looked into in depth in Biology 1. So we will cover the cytoskeleton in depth in its own lecture. We have the plasma membrane, of course, so every cell has its own outer membrane to keep it independent from the cells around it. Okay. And we have the cytosol, which is the jelly that fills up the cytoplasm. Okay. So in the class that goes along with this lecture, I'm going to have you guys work with these organelles. So you're going to look into the organelles. I'm not going to talk about them individually here. You guys are going to do that work. Okay? But these are eukaryotic structures. Most of them are membrane bound, and that is what gives eukaryotes their specialization. There's all these cool things. Cool? So, next topic is the way that each of these types of cells reproduce. So one of the qualities of life is that the thing must be able to reproduce itself, otherwise it's not alive. We've already clearly stated that cells are alive, so they must be able to reproduce themselves. And so, at a quick glance, ultimately, the goal is the same. I'm starting with one cell, and I'm ending with two cells. Okay. So, on the surface, same stuff. Right? I'm just making more of the cell. Now, because prokaryotic organisms are simple, and more specifically because their DNA is simple, it's that singular circular chromosome, their reproduction process is a lot more simple, right? What they do is called binary fission. Binary, TUI. Fission means to split apart. And so it's a rather simple process. The bacteria is like, yo, it's time to reproduce myself, right? Whether it's because the conditions around it are good, because the conditions around it are bad, whatever environmental triggers, like, yo, bacteria, time to reproduce, man. All right, I'm on it. First thing it does is it grows, like twice its size, it gets big. And then it copies its circular chromosome. And so there's a little area inside of the chromosome called a point of origin. And the reproduction of or rather the replication of the DNA starts there and then moves along. Right? And so it reads it and reproduces it. So you get these budding circles, right? So you have your original chromosome and you have this new chromosome coming off of it. And it goes around and reads it and reads it and reads it and reads it. And then by the time it gets back to the end of the circle, we click back at that point of origin, and then the two circular chromosomes can be spliced apart. And now I have two copies of the DNA. And then the bacteria itself cuts itself in half with like a protein lasso. Right? So if you imagine, what's something soft, a marshmallow, right? You take some fishing wire, you wrap it around the marshmallow, and you pull it real tight. They pop off into two marshmallows, right? That's what this bacteria is doing with this protein filament that's wrapped around it. Tight, 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 pop. Okay. And it pops off, and now you have two bacteria instead of one bacteria. Okay. Genetically identical, asexual reproduction, very fast, very efficient. Right? And they can do this because they are this single-celled, simple organism with this circular chromosome. Nice, clean, easy peasy. Binary fission. The eukaryotic evolution of that same process. And so a lot of the proteins involved in binary fission show up in mitosis. So mitosis is an evolution of binary fission. And so one of the ideas that we have to keep in mind when we look at cells and comparing the cells and just life in general throughout the course is how do we see evidence of evolution through the maintenance of mechanisms and chemicals and proteins and qualities. And so this is one of those things that we can look at and say, oh yeah, eukaryotic organisms definitely evolved from bacterial organisms because they share the same mechanism, they have the same protein doing the same thing. Right? And so the eukaryotic cells have a much more complex structure of genomes. Right? And so our chromosomes are linear and they're individual. So I can't just start at one point and copy and pop it off. But I do need copies of all of my chromosomes. And so mitosis is the evolved version of binary fission. It has the mechanisms necessary to make sure that I copy each and every one of my chromosomes, and I copy them correctly. And then I evenly split them into two groups. I surround those two groups back in their nucleus, and then I can cut my cell in half through cytokinesis, and now I got two cells. Right. And so, Matt, you should be familiar with the phases of mitosis from Bio 1, but that's another thing that we'll cover in a separate lecture, right, and delve into it a little bit more in depth than you would have in your freshman year. We'll look at the movement mechanisms, right, because I'm sure they'll tell you how they actually move. Right? How do they make sure they're in the right place? It's not like they have brains or anything. Right? So how do they know they're in the middle when they're in the middle? That's what we're going to look at. How does the cell know it's time to do mitosis? Right? That's the thing we're going to look at. What if a cell wants to stop doing mitosis? What if it's a bad cell? Right? What if you are a diseased, dirty cell? Does your body want that cell to reproduce itself and make more dirty, diseased cells? No. Right? And so there are mechanisms that we have in place that regulate mitosis that are more advanced than binary fission things. So we'll look in all those things in its own lecture time. Okay? But binary fission, bacteria, copy the DNA, split it in two. Mitotic cell cycle, eukaryotic organisms copy their DNA, split into two. Okay? Cellular reproduction. So, on the board behind me, I have a very rough timeline of the evolutionary course of these cells that we see on the planet today. And now there are some points in this timeline that are contentious, that are still up for debate and are still theoretical, but I'll explain why I've put each of these points where I've put them. Now obviously we have to start with the first cell. So once upon a time there was no cells at all. We had this planet that was full of energy. There was like lightning storms and chaos. There was no oxygen, right? Our ozone layer wasn't made yet. So the sun rays were bombarding our planet, tons of energy everywhere, okay? A lot of heat. And so the molecules on the planet were just slapping against each other all the time. Right? And so I'd make a random molecule and it would fall apart. I'd make another molecule, it would fall apart. I'd make another molecule, it would fall apart. And this was happening billions and billions and billions and billions and billions of times. And eventually, through the course of probability and all those billions and billions and billions of opportunities, we got a combination of molecules that were able to self-perpetuate. So effectively, I got this little bubble, and inside that bubble, there was some molecule, okay, at that time it was RNA, that was able to copy itself and the bubble around it. And so as soon as I had that one self-perpetuating bubble, it could make 2 bubbles, it could make 4 bubbles, those 4 bubbles could make 8 bubbles, and it didn't have any competition. So it was just killing the game. And so as soon as that first cell appears, I have the beginning of evolution, and it just rolls on down from there. So this first cell was a prokaryotic cell. Super primitive prokaryotic cell. And so for a few million years, I just have my simple prokaryotic cells getting better and better. So every little change that made that cell a little bit stronger for its environment gave that cell an advantage, and it would copy itself more than the other cells would copy themselves. At some point in time, a prokaryotic cell got the ability to wrap its inside mechanisms in a membrane. Right? And so it's like, oh, your job is making bananas. And so I'm going to give you banana-y stuff and put it in this membrane, and now you can make the bananas for me. And so now that cell was better than all the other cells. Okay? That was our first eukaryotic organism. It was able to wrap its DNA inside of a membrane, which kept its DNA nice and safe, which is super important. So now I have eukaryotic organisms. Very simple eukaryotic organisms, but I got eukaryotic organisms. At this point in time, there's still not very much oxygen on the planet. Very little oxygen. But there are a lot of chemicals floating around, and these living things are using chemosynthesis to make their sugars, to get energy, to do their cellular work. And one of the prokaryotic organisms develops a mechanism that allows it to break down that sugar more efficiently than the other prokaryotic organisms can. And that process is aerobic respiration. That's using oxygen to break down the sugar to get better outcome. You get more ATP from aerobic respiration than you do from anaerobic respiration. Okay? So this is what we use today in our cells when we have oxygen available. Right? This is what drives us most of our ATP production. Now again, oxygen levels at this point in time were very, very low. So these prokaryotes that use oxygen are just using little amounts. And so the prokaryotic organisms that are aerobic today, and we look at them, in low oxygen environments, they do really well. And that is indicative that these mechanisms evolved in low oxygen environments. And we use that data to put this development of aerobic respiration in front of the development of photosynthesis. So we get some time between this development of aerobic respiration. Some prokaryotic organism all of a sudden is like, hey, I can harness the energy of the sun and use that energy to put together my sugar. A lot more effective than using the chemical energy to put together the sugar. So this prokaryotic organism can now do photosynthesis. And now photosynthesis, of course, generates oxygen. So I'm starting to oxygenate the environment at this point in time. And so this bacteria that can do photosynthesis is doing really well, making lots of copies of itself. Right? And we're getting more and more and more oxygen into the environment. Now, at this point in time, a very important thing happens, right, where this branch occurs in my timeline. There is an eukaryotic organism who envelops, wraps up, one of the organisms, the prokaryotic organisms that can do aerobic respiration. So you got this eukaryote, right? He has this mechanism for surrounding his DNA. He has some other little organelles doing organelle things, so he's doing really good, right? And he meets up with this prokaryote who can break down sugars using oxygen and gets a lot more ATP. And so he swallows them up. And now the prokaryotic organism lives inside this eukaryotic organism. And so this prokaryotic organism is protected. And it's good for the prokaryote to be inside of that. The eukaryote is benefiting from all this extra ATP production. The prokaryote is benefiting from the protection from the eukaryotic organism. And they undergo a process of endosymbiosis. So eventually this prokaryotic organism just becomes an organelle of this eukaryotic organism. It doesn't need its own mechanisms anymore. The eukaryotic organism is doing most of the work. So this one little prokaryote starts losing a lot of its mechanisms because they're just a waste of energy. It maintains its DNA, and it maintains its membrane, and it maintains its ability to do aerobic respiration. And this prokaryotic organism becomes the mitochondria that we have in our cells today. The evidence of this endosymbiotic process is that our mitochondria have their own DNA separate from the DNA of our cells. They have their own membranes. So the prokaryotic organism living inside the eukaryotic organism that could do aerobic respiration becomes the mitochondria. So I get this break in my timeline, right? And so down here I have my more simple organisms. They're the ones that didn't engage in this endosymbiotic process. Okay. So for the most part, these are what we get as bacteria today, our prokaryotic organisms. Okay. However, there do exist some anaerobic eukaryotic organisms that still persist in the planet. They're very rare, and they live in really weird places. Right? So they thrive in places where there's not much oxygen. And so they evolved under conditions where having aerobic respiration didn't benefit them at all. So they never did the endosymbiosis with the prokaryote that could do aerobic respiration. They never got the mitochondria. And so, where are they? They're very, 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 very rare. Don't see them very much, okay? But they do exist. And so they have to be talked about. Here they are, okay? Moving on. Back to our timeline split. Okay. So at this point in time, everybody up here is eukaryotic, and everybody up here has mitochondria. Okay. And so everybody has the mitochondria, they're living the good life, they're doing aerobic respiration, they're getting mad ATP, right? And then they're like, you know, it would be great if we could also do photosynthesis, right? So one group of the eukaryotic organisms, right, it would have been one individual of the eukaryotic organisms, right, who has a mitochondria, meets up with a prokaryote who can do photosynthesis. And the same process of endosymbiosis occurs. That prokaryotic organism moves inside of the eukaryotic organism, right, and it's doing photosynthesis. It's giving its extra sugars to the eukaryotic organism. eukaryotic organisms protecting the prokaryotic organism, giving it the resources it needs, right? And they're in this happy friendship. And then over time, the prokaryotic organisms lose their individual mechanisms that they don't need. They maintain their DNA, and they maintain their ability to do photosynthesis. And so these organisms, they become chloroplasts. And then so we have a break here. And so up on this is the plants and the algae who have chloroplasts. I should have wrote algae. I didn't write algae, but I should have. Algae is up here too. And so any of the eukaryotic organisms who have chloroplasts are now going down this branch of the storyline. And then this branch leads us to the other eukaryotic organisms who never gain the ability to do photosynthesis. So things like us, animals, things like fungi, right? And things like protozoa. And so these guys all have mitochondria, but they don't have chloroplast. So they can do aerobic respiration, but they have to get their sugar from somewhere else. Okay. So this is a story of cellular evolution and endosymbiosis. So endosymbiosis is one of those huge, useful things when you're talking about evidence for evolution. Because you have mitochondria that used to be prokaryotes, and you have evidence of that. And they still have qualities that the prokaryotes have. And so that's it for our introductory lecture on cells. Right? You should know a lot about cells already, so I don't think I have to talk too much about the basics. If you have any questions, of course, feel free to ask during our Zoom times. Again, we will be delving into the individual organelles during activities of the class. But yeah, cell basics, so the cell theory, all living things are made of cells. Cells are the smallest unit of life. All cells are derived from other cells. I have simple prokaryotic organisms that have no membrane-bound organelles. I have eukaryotic organisms that have membrane-bound organelles. And the membrane-bound organelles allow for specialization of mechanics within the cell itself, a concept called compartmentalization. And yeah, I think that's it. So I hope you've enjoyed this side of the classroom. I hope you have learned something, or at least had something refreshed in your brain hole from this lecture. And have yourselves a fantastic day.
Take me to the place where the white boys dance. Take me to the place where they run and play. My baby is gone, you might have a chance. Just take me to the place where the white boys dance. From the whiskey to the key. Take me to the place where the white